All right, team. We are doing a little bit of CSI this week, and our goal, um, if you start to click into our CSI Wildlife here, it is to identify the origin of an ivory sample that has been seized, right? So there's been poaching that's occurred, and we have this ivory, and we want to figure out where in Africa this poaching occurred. These little green sections are all of the places where wild elephant populations live today. And we can use some DNA analysis to do this. So in general, as you're working through this, you want to watch every video when you see a place to watch. They're short videos, but they're really helpful. You want to read all of these blurbs where you see a place to read. And what I'm going to do here is I'm just going to get you started on case 1. I'm going to give a little background, and then you can work through the analysis of case 1, do case 2 on your own, and wrap up and make sure you understand with our short practice quiz on Canvas, okay? So in general, we can see that we've got a couple elephants here, and we're using some kind of technology. And it turns out that what we're doing is something called DNA profiling or DNA fingerprinting, even though it doesn't have anything to do with fingerprints, but that's the term that's used. And what we're really doing here is we're taking advantage of these things called short tandem repeats, which is a section of a gene that has really variable or really different versions in different individuals, and it has to do with how long the piece of DNA is and how many times a little chunk is copied. So let me show you what I mean here. If we have this section of a gene here that you see in this example, it starts off with this AAGT, and then look what happens here. We have a repeated unit of just a few bases. In biology, we use bases or base pairs to mean the letters in DNA. So just a few letters here, CTA. It happens again, then again, then again, then again, then again. It happens over and over and over. It gets repeated. So that's where this repeat word comes from. And the thing is that's really cool is that it gets repeated a different number of times in different individuals, which then as a result means that different individuals have a different length of this gene in total, because some of them might have 9 repeats and some of them might have 14 repeats and some of them might have 2 repeats. And so we can identify which individual a DNA belongs to just by figuring out how long it is and how many repeats they have sometimes. So what I'm going to do is I'm going to back out of here. We see that we're using this technology in a couple of individuals, and somehow we're able to visualize the differences. And I'm going to show you sort of in a simpler setting what we're looking at, and then we can come back to our elephants. So again, short tandem repeats are these really variable or highly different regions in our genes. And remember, genes are just these short segments of DNA that code for proteins, which correspond to our trait. And a short tandem repeat is the 3 to 5 letter or base pair sequence that repeats a different number of times in different individuals. So the last thing that I want to throw out there, and then we'll take a look at this much closer, is that different individuals, whether that's people or elephants or anybody, we can be either homozygous, which means that we have the same number of repeats for this gene in the copy that we got from mom and the copy that we got from dad. So we can either have 2 of the same allele with the same number of repeats, or we can be heterozygous, which means that we got a different number of repeats in the allele from mom compared with the allele from dad. Let me show you what that looks like. Okay, so imagine this is me up here, and notice that there's 2 rows here because I have an allele from mom for this gene, and I have an allele that I inherited from dad, and I would be considered homozygous for this gene Because both of my alleles have the same number, have 2 copies of the short tandem repeat. Now let's compare that to you down here. You would be heterozygous because your allele from mom has 3 copies of that STR, and the allele from dad has 5 copies. So your 2 alleles are different lengths because they have different number of copies of this STR, right? Now, let's say that this is you down here, this heterozygous individual. Just to illustrate that, if you're like, what are you talking about from mom and from dad? Each individual has 2 copies of each gene, and they pass on one of their copies to you. And so you could have a homozygous parent that has 5 and 5 copies, and a heterozygous parent that has 12 and 4, and you would inherit one allele with one of their numbers of copies from each parent, right? So quick question, this is the offspring. Are they homozygous or heterozygous? They'd be heterozygous because they have different numbers of copies in their allele from mom and their allele from dad, right? Okay, so the last thing that we got to add is this idea of a flanking region. And the flanking region is just the chunk on either side of our short tandem repeat region. And what I mean by that is we see here that we have 1, 2, 3, 4, 5 copies of this STR region, the CTAT. And on either side, we just have some other letters. And that would be the surrounding portion of the gene, okay? Now notice that even though this copy on the bottom, this allele has 7 copies of the STR compared to 5, it has the same letters in its flanking regions. Because we're looking at a single gene, and the only part that's different is how many times this little chunk is copied, okay? Now, how do we end up seeing those differences? DNA is microscopic, and we can't just like count the number of copies of the STR. So we have this technology called gel electrophoresis, and this is really cool. What we have here is actually an electrical box. And it turns out that DNA has a negative charge. And so what we can do is we can take advantage of the idea that positive and negative charges attract, and we can load our DNA samples into these wells or the holes in our gel, and the DNA will move towards a positive electrode, which we can put at the other end of our gel. Now I say gel because this truly is a block of what kind of looks like Jell-O, and it has tiny little holes in it that the DNA can move through, okay? So we load it on one end, we put a positive electrode on the other end, and then we turn our electrical box on, and the DNA moves through the holes in the gel towards the positive electrode. Now, we see these different bands because we had different numbers of STRs, right? We had some alleles with only 2 repeats, and some alleles with 3 or 5 or 14 repeats. And so what happens is the bigger, longer segments of DNA that have more repeats, they don't move very far from the well where they were loaded because they're big and it's hard for them to get through those holes. But the tiny little fragments, like the ones with 2 STRs, they travel fast, and so they go farther down the gel in that same amount of time. So we can separate DNA molecules by their size or by the number of STRs, okay? Now, the last thing I want to show you is how we're going to use this. So the general idea, we look at a gel like this where the wells are on the top, and we call the column a lane. So we say, this would be lane 1 on the far left. I loaded my DNA here, and it runs down the gel toward the positive electrode. And like we just said, the bigger fragments of DNA move slower, so they stay towards the top. But they travel further the smaller they are, okay? Now, let's go back to our elephants. So what you see here is 2 different elephants. And we have many bands on this gel because we can look at multiple genes at a time. And we have a DNA ladder on the left because we can load or preload segments of DNA where we know how long they are so that we can have those known lengths and use it as a reference to figure out how long our DNA segments from our samples are. Okay, so there's the background. Let's keep clicking through. There's a great video to watch here on technique. You can see how we would run the gel, time would go by, and these little segments of DNA separate out by how long they are. So quiz yourself here. These would be the what? Short or long DNA segments? I'm going to reset it. These are the short ones that move all the way down here because look how fast they move, and that's because they're smaller and they can get through the pores of the gel. And we can check ourselves because over here it tells us that down here this is 60 base pairs or 60 letters, and that means that this is a shorter segment of DNA down here. Okay, so moving through here, what we can do, and the reason that you saw so many bands for each of these elephants at the beginning, is that we can look at multiple different markers, okay? So we can look at, in other words, multiple different genes where the elephants vary in the number of short tandem repeats they have. So for instance, I could look at marker A, which is one gene, and like we just talked about, if an individual is heterozygous, I'm going to see two bands appear because they have two different versions of that gene with two different numbers of short tandem repeats. Whereas this individual must have two of the same allele, they're homozygous, because I only see one band. And I could compare these. I could look at a second marker, a different gene, and now this individual is heterozygous and this individual is homozygous. And the more markers I look at, the more DNA I have to compare to a mystery sample, like the DNA from a tusk. So let's keep going through. Let's look at our review section here. And remember that we can figure out how long a DNA band is, how long that segment is, by looking at the ladder. So let's review here. We've got a question that says the band in lane 1 has the following sequence. It's 41 base pairs. In this example, it consists of 9 bases, although in reality the flanking regions are longer. The 8 4-base repeat units. So we've got 4 bases in our repeat. It looks like it's TGAT, or no, we've got GATA, 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 GATA. You see that? So that it says that happens 8 times for a total of 32 bases. And then the flanking sequences consist of 9. So there's our 9. So we add 32 plus 9, and then that gives us 41 in total bases in this example here. So it's asking us, how long would you expect an allele to be if it had 10 repeat units? So 10 instead of 8. So we would take the same length, and if it had 2 more repeats and each one is 4 base pairs, then we would add 8 to that. So I'm going to say 49. And it says the fragment corresponding to the 10 repeat allele, what rate is it going to move down the gel? Is it going to be faster or slower or the same rate? Well, if a segment is longer, we know it doesn't go down as far as fast. It's slower because it's harder for it to move through the gel. Yay, we're right. Okay, so now you've got to analyze the DNA here to solve the crime. And what you're going to need to do is look at each individual marker and compare each elephant to the mystery DNA taken from the tusks, right? So here we have 6 different elephants, and looking at marker 1, you want to compare where these bands fall to see who matches the elephant right here.